Welcome everyone to our 18th webinar in Contact's Produce Safety webinar series. Today we finish part 2 of root cause analysis. This webinar series continues to tackle fresh produce safety topics every month, where we bring together industry, academia, and regulatory minds to solve some of the produce industry's most timely challenges. This series is part of a larger industry outreach and risk mitigation project called Contact, or scientific challenges and cost-effective management of risks associated with implementation of produce safety regulations. This project is supported by a Specialty Crops Research Initiative grant from the USDA National Institute of Food and Agriculture. Any opinions, findings, conclusions, or recommendations expressed in this presentation are those of the speakers and do not necessarily reflect the view of the U.S. Department of Agriculture. Contact is made up of over 10 universities and government institutions. And if you like numbers, that's over 50 faculty, staff, students, and volunteers, not to mention the support of our Produce Advisory Committee, our PAC. While we understand we can't list all the names, I hope you get to see their logos. It takes a village, and I thank each and every one of you. Also, in my follow-up email with the webinar recording, I will provide information on how you can serve on one of these USDA Specialty Crop Research Initiative grant panels, where your voice can be heard and you can direct research and extension projects like Contact. This year, we'll begin each webinar with an update about one of Contact's 7 research or outreach objectives. Today, we are going to highlight some of the work from Objective 4. As a reminder, Objective 4 is focused on post-harvest washing and transfer and survival of foodborne pathogens on food contact surfaces. Here, we are highlighting some work from the University of Georgia that demonstrates the benefit of applying sanitizers during post-harvest washing. In this work, water with varying levels of organic load, shown as black bars, for 300 ppm and red bars for 2,500 ppm chemical oxygen demand were treated to achieve residual of 20, 40, and 80 ppm free chlorine and peroxyacetic acid, and compared to a no sanitizer treatment. A single inoculated cucumber was washed with 9 other uninoculated cucumbers and transferred to the water, and produce was determined. Shown on the y-axis is a log 10 transform value for percent transfer. A value of -5 would equate to needing more than 10 million colony-forming units of salmonella in order to observe transfer to other cucumbers. As you can see, there was significantly less transfer in systems with 20 to 80 ppm of either sanitizer compared to the untreated controls. This should tell you, add a sanitizer, it's important and it's working. There also was not a significant difference in performance between COD levels. We could definitely dive into this deeper, but this is just a small look at some of the findings generated from Contact. So stay tuned to hear more from the other teams. This year's webinar series will always address topics chosen by you. Don't forget to quickly tell us your thoughts in the survey evaluations as you exit the webinar. We've got an exciting list of webinars scheduled for this year, covering topics like energizing your employee training programs and keeping them fresh, and also sanitation at retail. Be sure to check out the Contact website and YouTube page for more produce safety resources, including our key takeaway documents, which summarize the really important highlights from the webinar, our webinar recordings, and much more. Subscribe to our page to get instant notifications of content like our mini webinars. Okay, a quick reminder for today. If you would like to submit a question for our speakers during or before the Q&A session, please submit your questions through the Q&A box and not the chat box. And now let's welcome back friend of Contact, Dr. Jennifer McEntire. Dr. McEntire is the founder of Food Safety Strategy. She's a food microbiologist by background and has worked to bring scientific perspective to food safety regulatory issues. Dr. McEntire has held several food safety leadership positions, most recently with the International Fresh Produce Association. So, welcome back, Jen. The floor is yours.
Thank you. We can move on to the next slide while I introduce the other panelists today. Laura had brought up at the beginning the menti.com. Wow, you all are really on top of it. That's fantastic. For those of you who are just joining, if you go to menti.com and use the code, please identify your role in the produce community while I'm reading the bios of our wonderful panel today. First up, we will have Vivian McCurdy, and she serves as the Division Director of the Food Protection Division for the Indiana Department of Health. She's passionate about food, including the farm-to-table local movement. And in her career, she's worked with small growers as a GAP auditor, and then served as the produce safety manager in implementing the produce safety rule for Indiana. After Vivian, we'll hear from Felice Abrassier, who abandoned her dreams of becoming a professional ballerina to become the Director of Food Safety and Quality Assurance Supply Chain North America for Dole Fresh Vegetables. She's worked in food safety and regulatory compliance for over 15 years with a wide variety of experience, wide variety of products that she's covered, most recently produce. Then we will have the birthday girl, Laura Strawn. For those of you who missed Alexis's introduction, it is Laura's birthday, and she's choosing to spend it with you here on this webinar. Laura is an associate professor and extension specialist in the Department of Food Science and Technology at Virginia Tech, and her research program focuses on the microbial safety of fresh fruits and vegetables. She does do some work troubleshooting various food safety challenges, and we look forward to hearing from her today. Then to wrap things up, we'll have Dr. Tim Jackson, who's serving his second term as IAFP president, but his day job is as a senior science advisor for FDA, a role that he's had since last year, joining in 2022. In this role, Dr. Jackson supports programs in outbreak investigations, root cause analysis, and prevention strategies. So it looks like, not surprisingly, we have a wide variety of participants, attendees here on today's webinar. A lot of people on the inspector regulator side, but a great representation from all. And so we can go ahead. We've got one poll question for you before we kick things off. Just your opinion, no right or wrong answer, but who is best positioned to do a root cause analysis? Hopefully many of you joined last month when we were talking about some of the basics. Today, we're going to get a little more hands-on into the how-tos. But curious whether you joined last month or not, what do you think? Who should be doing a root cause analysis? See a majority who's saying that it's a team of industry, several from of the above. The industry is getting nearly a quarter, with a couple of you voting for some different areas, extension, regulator. Little curious as to what the other is. And so if anybody wants to volunteer, who else they're thinking about in terms of roles in doing a root cause analysis, I'd be curious to see that. We'll give it just another couple of seconds because I think we've stabilized here. And then without further ado, let's go ahead and get started. Vivian, I will turn it over to you.
Good afternoon, everyone. Thank you for having me today. So first of all, when we talk about how to conduct a root cause analysis, one of the most important things is understand how each element contributes to the whole situations. And what we're talking here is about the when, the who, the what, and the where. And then once we have the data and analyze it, then that is the time that when we can implement some possible solutions, and based on the solutions and the finding, and then we can evaluate our result and maybe fine-tune even a better solutions from then on. Next slide, please. So first of all, what is the problem? Oh my goodness. So that is something that we often, when we having issues, we have more questions than answers. Everyone say, oh my God, that's a problem. But really think about, break it down into different elements. First of all, is it a biological problem? If it's a pathogens, what kind of pathogens are we talking about in here? Is it Cyclospora, the new kids around the block? Listeria, Salmonella, E. coli? Is it physical? Is it chemical? Is it a pesticide causing the problem? Is it the sanitizer is the one that causing the problems? And also, what are the vehicles of contaminants? We know about the 6 big issues with produce that are more likely to cause foodborne illnesses, the high-risk produce, but that could be something else too. So don't forget that. How about systematic? Is there a one-off incidents? And if it's systematic issues, that is the time we really need to go back to historic information. Do we have access to any WGS informations? Can we talk to our state partners, the lab people, and say, can they run something for us and post all the data off on pulse labs to help us to understand what's going on? Is it seasonal? Is it only happen when during the summer? Is it location? Is it the special regions? Again, a lot of questions, very little answers. So that is the next thing that we need to look at it. The next slide, please, is where to find the problem. Now, so in produce, we always say 1 step forward, 1 step back. Again, it's very systematic way to think about it. Do we go all the way to the seed? Is there something wrong with the seed? Does the seed come with the pathogens? Is it all the way to the harvesting, the packing? Is there something wrong with zone 1? Are we getting contaminations from it? And then you have to be very objective when we're going to find where the problem is. The thing is, when we try to do our best, and I know that many growers, almost all the growers we encounter, they don't want anyone to get sick. So when we present a problem with them, we all have to be very objective and say, okay, this may not be likely the top 6, but that could be it too. So let's don't rule out anything until we do the data analysis. So inside the facility, what is the zone 1? How does the zone 1 maybe the possibility of contaminants? Is the pathogens there? And then zone 2, zone 3, and zone 4. And sometimes when there's a multi-state outbreak, we really need to look at, do we need to do some supply chain analysis? Do we need to do traceback analysis? And if we do, that is going to be a lot more work, and which means that we have to go and talk to the patients who are sick in the hospital and ask them where do they get the food, and then go back to the grocery store that they purchased the food, and then go and so on and so forth. And again, as I said it earlier, systematic is the key. And when we know where to find it, the other things that we really cannot ignore, especially in produce, is environmental issues. Is this some weather patterns that we know that was a flood happened during the growing season? Is it in areas that's just more prone to more bird, more animals right around the area? Those are the things that we need to look at as the where to find the problem. The next slide, please. So now we have the what and the where, and now it's really to think about how to collect those data. The most important thing when we do to collect the data is really come up with a plan, a data collection plan. And with the plan, we need to know who are going to be there to help us out to collect. Who's our team? And do we know who is going to do what and when and why? Do we know that everyone's the communication method? Especially when we involve growers, we talk about the industry should be included in the root cause analysis. When we talk to the growers, we need to understand, are they familiar with digital, electronic communications? Is this something better off to be doing it face-to-face? It may be a combination of both, but the most important thing is bringing the subject matter expert, your local extension office or the FDA CIFSAN, and they are very good and they have lots of knowledge behind the science of produce and the science of foodborne outbreak. So make sure you bring in those people and need to be part of your team. And then really down to the data collection methods, the next thing. As I said earlier, it can be very overwhelming, right? You have a lot of leads, you don't know which one to start with. So deal with one theory at a time. Pick the easiest one first. Talk to your subject matter expert, talk to your growers. And you have to be, when you do the data collection, you have to be situation appropriate. You're not going to do some soil collection when you think it's a water problem, right? That's just easy. And timing is the key. When are you going to collect the data? During the harvesting, the growing, the packing? or during the transportation, or it's actually when you reach out to the retail store. Those are the timing issues. And one thing I want to emphasize again and again with my team and with everyone else, make sure that when you collect the data is using the aseptic method. The last thing you want to do is contaminate your own data. Next slide, please. I have a few examples in here. First one is the water testing. As I say, which one would you use? If it's something that you know is water, is from your well, then you're probably going to do the faucet method. But if you think it's an environmental issue, something leak out from somewhere that going into your water, your well system, then you probably want to look into the dead end ultrafilters method. It's a lot more expensive, but it gives you a lot more information. And then down to where to swap. When we look at facility, where is the food contact surface area? Where is the zone 2? And sometimes there's a couple times when I go in to visit a farm, there was a pallet and someone stepped on the soil and then stepped on the pallet. And then before I know, they were putting the product on the pallet. So that is where you will swap. And that is really what I say, be objective and pay attention. What is around your area? Environmental assessment. The picture on the left is actually a turkey farm right next to one of the growing plots that I was working with. And the one on the right-hand side, and they were spraying. And what are they spraying? We don't know. And those are the things that we need to find out. And the last thing is very important is documentation review. There's a P in the farm because we want to know what have been done and when and where. So when we do documentation review, really try to highlight what have been done right and what have been missing. For those that are missing, really go back and ask the workers and say why things are missing and how it was recorded and what have been shared with other people. Next slide, please. Now we have all the information, documentation, just like an SOP. If we do not document what we collect, we will have no idea how to do our data analysis. And when we do documentations, don't be afraid. We talked about it last time. People do not like to put things in writing because they worry about how's the information going to be used. But please don't. When you do root cause analysis documentation, it really is the key to give you information. Because one-off event for this year could be a multi-year incident. And if that's the case, we really need to go back to the documents and find out what have been implemented as a solutions and how we're going to continue to fine-tune and find the problem. So inside the documents, everything have to be detailed. Don't be afraid to be over-communicate. Be objective, descriptive. If you have lab result, WGS, make sure you include those two. Don't miss out on that. And then if you did any collection error, write it down. And that is how we learn, and that's how we're going to get better. And now I'm going to pass on to the next presenter and give you guys more examples explaining how to do root cause analysis.
Hi, everybody. We can go on to the next slide. So thank you, Vivian. I think Vivian teed it up really nicely for me. I'm going to be talking to you about more real-world examples from a company like Dole and how we do root cause analysis when we encounter a problem that doesn't necessarily involve regulators or the entire industry, but how we do it internally. I started off with this slide just as a kickoff slide. I think many of the presenters in the previous webinar have gone through this, so I'm not going to go through the definition and the purpose, but it is something that you can easily find through grandfather Google, and he can give you a nice definition of root cause analysis. You may encounter sometimes where you need to talk to the business of why you want to do root cause analysis or why you maybe need to develop a process or a procedure around doing root cause analysis. And so I just wanted to give you some talking points. It does address issues at the source. It does help improve decision-making, reduces errors, save resources, and then also shows your commitment to improvement. So you can use those as potentially when you're explaining things to your leadership teams on why you want to develop a program if you don't have one just yet. Next slide, please. The way my mind works, and maybe some of yours, is I have to have kind of an outline when developing a program, and then I can get creative on how I'm developing that program. But root cause analysis process steps, they seem easy enough, right? Not so much. There's a lot of resources out there, and I think this webinar has done a great job with providing a lot of those resources, but when you get into it and you're trying to develop a program or actually do one, it may seem a little confusing. I put in 8 steps to conducting a root cause analysis, and we call them root cause assessments here, but it's the same thing. And I'm not going to go through these one by one, because what I'm going to get into in the next couple slides is real life examples of things we've done here at Dole and how I've incorporated using these 8 steps. Next slide. So you may ask yourself, how do we get started? Perhaps you don't have a root cause analysis program currently internally at your companies, or perhaps you do and you want to enhance it, or you just need some ideas. So I thought I'd put together how do we get started, or at least how we did it here at Dole. So taking that very first step. So really, you want to determine what issues may call for a root cause analysis, and really have an open discussion with your team. Not everything's going to call for a root cause analysis. So you want to determine what are those things that right away we want to be able to go and do a deeper dive into. You may not complete your list, but at least you can have some starting points. And then strategize on the resources or resource you want to use. I know Vivian shared the fishbone diagram. There's the 5 why techniques. There's a lot of different techniques out there. So what do you feel comfortable using? And for the people that are actually going to be out there, boots on the ground conducting, what do they feel comfortable using? And maybe even develop a template for that. We built a digital template here at Dole. It can be used on a smartphone or on a tablet, and it's a series of questions to ask to get you started down the path of the root cause analysis. And I'm going to approach the first example from the field. So this series of questions, when you go out into the field, you can start asking and documenting, and then also taking different types of pictures and adding additional comments to that as well. They can be useful because then you can look at your data holistically, season after season or year after year, once you start collecting that data. Also interviewing operators and asking a lot of questions. When you're out there, no question is ever a dumb question, so ask them all. And here's where you can probably incorporate the 5 whys. Why did this happen? Why did we do the different types? And I think that's where we incorporate it a lot when we're doing these ourselves. And then while you're conducting your assessment, making sure you're taking notes of anything that may be out of the ordinary or any contributing factors that you're seeing out there. Next slide. So here I'm going to get into an example. So this example is not just one. I took a bunch of things across a couple years and put it into one example to share with you. So example number 1, where we would get triggered to go out and do a root cause analysis. Lab calls at 2 o'clock in the morning and tells me, Felice, you have multiple salmonella positives across lots on the same ranch. So if you go back to that slide or remember those first 8 steps, they're here listed. So what was the problem? The pre-harvest test came back with multiple positives. So now we're going to go collect our data. So within 24 hours of notification, my team knows that they have to go out and start the root cause assessment. So they're going to take that digital tool and they're going to go out and they're going to start asking the different series of questions that are on that tool. They're going to be taking pictures of anything noted. They're also going to be taking additional samples if they deem it necessary. So it could be additional tissue samples, water samples, soil samples, if there's soil amendments present, things of that nature. Then they're going to go to the 3rd step, which is identifying the possible causes. They're going to be looking for contributing factors. So in a case like this, where we had multiple positives, they're going to go out to the fields and look at, do we have animal intrusion? Is there anything going on with the water sources? What is the adjacent land? Any kind of soil amendments? What were used? And then you want to interview your cross-functional partners. It could be your ag operation teams. It could be the grower. It could be the different applicators. It could be the irrigation people. A lot of different things. And then also taking note again, anything out of the ordinary, did it occur? Meet with the team once you get back and you've identified some contributing factors, you've taken your pictures, you've answered the questions, then you want to analyze that data. So you're going to come back, we're going to meet with the team together and we're going to go over what you saw out there. And then we're going to start to discuss, do we know the root cause just yet, or do we need to do more work? Identify the root cause would be the next step. So based on the data, Did we identify a root cause, or can we start developing more of a hypothesis? Then develop solutions. So maybe we didn't identify the root cause, but we found a lot of contributing factors. So did we identify any gaps in our process? Do we need to start finding some solutions to close those gaps? And then implement those solutions. Word of advice before implementing any solution, make sure you're having conversations with your cross-functional teams, make sure the solutions are practical, feasible. And then we use the info we found in this example to build a best practice document that we share internally as well as externally. And then the last step on that 8th step would be your monitor and verify. We would monitor the ranch for the remainder of the season and for future seasons, and make sure that if this was just an isolated incident, was it an issue with an input, or is it something maybe more we're not seeing on that ranch, so we need to be monitoring it season after season. So next slide. So here, I just wanted to give you some examples of what we saw and what some of the pictures were when we were doing this particular root cause assessment or analysis. So the team went out and took pictures, and some of these pictures you can see we have some soil amendments that are staged in close proximity to the ranch. You also see that where the dirt looks like there's tracks there, but there's a lot of animal tracks there. You see some different soil amendments there. And then you also see a picture where there's water and irrigation piping of the water. So what they noted in the report were that we stored uncovered soil amendments next to fields. They did take a positive, it was surface water. They took a sample of the water, it was positive. There were bird droppings in the irrigation ditch, things of that nature. Mold appeared to be in the soil. They needed to ask more questions about that. Animal intrusion, coyote tracks were observed. These were a lot of the contributing factors that were found in this example. While we were never able to identify the exact root cause, we did find multiple contributing factors where then on each one of them, we looked over our internal programs to determine, are we doing enough? Do we need to close any gaps? Do we need to add anything in here? And this happened with our growers as well as our cross-functional ag ops teams. And what ended up resulting out of this is we developed an organic best practice. And we learned a lot about organic farming through these examples. And so that's something that you can do as well. Maybe you don't find the exact root cause or the smoking gun, but you will find contributing factors. Next slide. Another example here, I did this one a little bit differently. We were alerted that a prohibited substance was found on lettuce. This was alerted to us by one of our customers. They got tested by a state regulatory agency, and there was a prohibited substance found. And so here are just some examples. I'm not going to go through all of them. I know we're probably going to share these slides, but some questions that you can ask as you're interviewing different people to try to figure out what happened. So in this case, what the outcome was that we were growing right next to strawberries. There was a very narrow roadway that divided the crops. As we went out there and started asking questions and looking at records and asking, hey, do you know about this prohibited substance, to our neighbor, they said, oh yeah, that's approved for strawberries. And as a matter of fact, on the day in question, there was a wind event. And so what probably happened is there was some sort of a drift. So we ended up taking some samples from the edge of the field that was closest to the strawberries, and we did find that prohibited substance. So really what we got out of this was get to know your neighbors, be able to talk to your neighbors, really ask a lot of pertinent questions. Remember, no question is a dumb question. And on this one, we were really excited because we were able to find the root cause and be able to provide a good corrective action, which was when dealing with our neighbors, really understanding when they're spraying, if there's a wind event, please don't spray, things of that nature. So this was a really interesting one. Next slide. So here, words of advice, if I may. I've done a lot of root cause analysis myself, and there's a lot of experts on here that have a lot of really good information. I always like to take the think outside the box approach. Sometimes in food safety, we can be very robotic. We have a lot to do, a lot of forms, a lot of documentation, but it's really important that you're thinking about what could have happened, what happened that was out of the ordinary. Use your senses, vision, smell, hearing, touch, and your intuition. Does something just not seem right? Don't taste anything. I know that's one of your senses, but don't do that. And then here again, just summarizing some of the things I talked about. Identify the type of resource you want to use. If you can digitize, do that, it helps. Use previous data to identify opportunities. Pictures speak 1,000 words, but also make sure you're commenting on those pictures. interview your operators. Your operators will always tell you where your failures are. They're the experts, but also be very well versed in what you're doing. And there's a lot of resources for you to become well versed in food safety if you're not already. Look at data to tell the story, and then learn from it. And if you can, incorporate it into your programs. Last slide. And on my last slide, I just like to put just breathe, because I know when you're doing root cause analysis, it could be because something wrong happened or what you perceive is wrong. And so it can rile up some emotions, or you could get anxiety or things of that nature. So it's just really important to take a step back, breathe, think about things, work through it. And more than likely, if you can't find the smoking gun, you are going to find those contributing factors that are going to help minimize that risk and mitigate things for the future, and move on with your day or month or however long it's going to take you to do these things. But thank you very much. And I didn't put a slide with my contact information, but happy to share that. And now I'll pass it over to the next speaker. Thank you.
Wonderful. So the reality of root cause analysis, of course, this is coming from an extension specialist, but I'm going to call this a practical case study and not giving up and embracing that you may never know, but you are better than you were before, and you've implemented a heck of a food safety program and a food safety culture. So I'm going to tell you a little bit about the Delmarva and some Salmonella Newport outbreaks that we had to deal with, tell you about all the things that we've done. And I think overall, it's a really neat story. So I like to think of root cause analysis as a good mystery. And like all mysteries, we're going to set up our characters, our resources, setting, plot, clues, distractions. Maybe you get to a conclusion, maybe it's satisfying, or maybe it's not. But again, if you want to think about root cause analysis, this is a really great way to do it. And truly, anybody can do this. You just have to be curious. Not to give everybody a geography lesson, but just a quick dinner party fact. What is the Delmarva? I know we have viewers from around the globe. This is Delaware, Maryland, and Virginia. It's a large peninsula on the east coast of the US. And what's so special about it is that it sits between the Atlantic Ocean and the Chesapeake Bay. And it's, other than a stunning place to live, and I lived there for 6 years, it's also home to some really great produce production, poultry, seafood, as well as recreational and beach area. So it's got a lot of things going on in it. And unfortunately, like I mentioned, it has had to deal with some tough events for food safety. We've had several different Salmonella Newport outbreaks over the past 20 plus years. So starting in 2002, and as I said, as recently as 2014, the map shows Newport cases for a specific fingerprint, this fingerprint 61. We can obviously see there's a hot spot surrounding this Delmarva area. And we've had 5 confirmed outbreaks. They were all linked back to tomato. We've had 3 other ones that, maybe not, but they also were with tomato. And then our 2014 S. Newport outbreak was linked to cucumber. So let's dive in a little deeper. Where do we begin? So I just mentioned this is a really long, almost 20-year process, but by about 2007, we were all sick of it. And so where do we begin? Like I said, in all great mysteries or root cause analysis, we're going to assemble that team. And here we assembled our team that had FDA, CDC, USDA, Delaware Department of Ag, Maryland Department of Ag, Virginia Department of Consumer Sciences, and then all of our extension folks, and our industry and our trade associations. We had this ridiculous diverse team, but it's so important because you have to bring fresh eyes, avoid biases, and really check and balance each other. So with anything, once you've gathered the team, we're going to start to get the clues, right? And this is where we identify that list of contributing factors. And we've talked a lot about different ways to do that, from fishbone diagrams to the 5 whys. And of course, I'm going to tell you a little bit of a different way that we did it. But the point is, it doesn't matter. You want to start your list. Then, of course, we want to collect our data and our information. And Vivian did a great job, and same with Felice, of showing how you can do that with different documents and apps. And then from there, we really want to come up with these implementations, right? Of how are we going to come up with these best practices? Because we need to be able to fix this issue, right? And then, of course, we want to collaborate and discuss all of our findings. But again, we really want to make sure that we stay nimble. So I'm going to show you that we had sort of two root cause analysis, one that began in 2007 and the other one in 2014, 2015. And I'm happy to share the outputs of these as the documents show. Like I said, lots of people do fishbone diagrams. We did not. We did a version of it, but we had all of these different flow diagrams, and you can see them talk about farm factors, farm landscape factors, management factors, transport factors, handling and packing. We came up with everything that we could. And do not stay rigid to this. This is just one way to organize it. Trust me, we had a lot of scribbles and things that we were inputting as we saw different observations and different conversations with different people as we were going through the process. Like I mentioned, let's start with where we got in our first root cause analysis and which factors we identified. We started off and we came down to these four big factors that were identified in this 2007 Virginia Tomato Food Safety Initiative report on what was going on in the Delmarva. And you can see a lot of them have to do with water. Remember, this is back in 2007. This is pre-produce safety rule. That was 2015. Pre-Florida tomato gaps, which became law in 2008. In Virginia, we somehow have a lot of shared growers with Florida. And so we pick up or adopt a lot of their practices. But you can see we identified pesticide spray water quality, irrigation water quality, lack of backflow prevention devices during our water usage, and then our one post-harvest water one, which had to do with flume water management. So what did we do, right? So we identified all of these different things from our list, crossing out what it definitely couldn't be, came out with these four practices of, hey, this is what this could be. And so from there, we decided to say, all right, let's really work with our industry. And our industry was really working with everybody else to figure out what are we going to do. So we implemented these several best practices, and you can see that it's never just one thing. It's not, oh, I can just put a Band-Aid on it, make it go away, it's good. It's a lot of different best practices and steps to really get in at these three pre-harvest factors that we identified with water: movement towards lower risk water sources, movement towards lower risk irrigation practices, a lot of drip under plastic, addition of sanitizers, right? So we were already starting water treatment, right? Either before or after our sand filtration systems, moving towards potable water if we weren't going to use water treatment, making sure also that we were going to use potable water during special phases. So during transplant, when we might have injury or damage to those root systems, and even during pesticide application, again, where it's going to contact that fruit. We also upped our water testing programs. This went beyond good agricultural practices, and then our regular maintenance performed and installing backflow prevention devices. A lot of times we're filling up ponds from well water and making sure that we have those backflow prevention devices to prevent contamination. On the post-harvest side, here we really focused a lot of our SOPs and new practices around management of post-harvest water. So this was monitoring our sanitizer levels during operation, focusing on infiltration prevention. So this is monitoring temperature of fruit coming in with our water temperature, making sure we had a monolayer, reducing contact with the water. And again, really making sure we had a way to keep those sanitizer levels at their right ppm throughout operation. Now, of course, because life is just life, we were sitting back, we were feeling really good. It was very all quiet on the Salmonella Newport Delmarva front. I thought I could go on vacation. No. And instead, we all ended up gathering the party back together because in 2014, we found ourselves with another Newport outbreak, but this time with cucumbers. And we actually really quickly as a team were able to go through using those old documents and all of our documentation that we had collected since then in the 7-8 years to figure out the water factors here really probably weren't what was causing this, or this wasn't a big thing. And instead we landed on one biological soil amendment of animal origin factor. So working with our investigators, working with our industry, figuring out that potentially we had an issue where unamended poultry litter was applied within 120 days of harvest. And so we were pretty sure that this might be a contributing factor. And again, it was probably likely not those water ones this time. So again, focusing in on what can we do and what were we going to implement of our best practices. Here we really focused on recommending or emphasizing use of synthetic fertilizer blends or composted manures, making sure we had really good documentation for certificates of analysis from our suppliers, following those USDA National Organic Program guidelines, very similar to what the Produce Safety Rule is recommending right now until we wait for FDA to come out with a standard about untreated manures, understanding that risk or really avoiding use. And we had a lot of growers that just decided, hey, we were just not going to do it. So like anything, and in the background of all of this happening, and I spent a lot of time focusing on what the industry did, because I think that's the really important part, right? But we also had a lot of close collaboration with industry to answer these questions, these longer research questions. Same with FDA, working with research labs to say, okay, where and what, how can we better understand what's going on here? So longitudinal and lab-based studies around water sampling, water treatment, salmonella survival in these different biological soil amendments, wildlife studies. So if we figure maybe the water's contaminated, how is it getting contaminated? Is it wildlife? Is it something else? Is it adjacent land use from poultry operations, seafood, preserves, recreational areas? We've had several long spanning projects to answer these questions. And if you're familiar with Center for Produce Safety, they also do something very similar with their rapid response. So industry is going to take immediate action. And then from that other side, the government or the research side, how can we answer some of these longer term things to be able to inform better best practices? And again, understand those sources and routes of contamination. We always want to promote awareness. This is a big part of any root cause analysis, or it should be, because the people who are in it, they definitely get it and understand it. But then you have to think about, what about the people who are not? And you need a communication plan to do these learnings and implement specific trainings based on this. So we spend a lot of time with an awareness of the situation, the history. We never want to forget our history, then we'll be doomed to repeat it. And we certainly don't want to do that. We really want to highlight our current best practices for prevention around Salmonella Newport contamination, our partnerships with industry, extension, government, gathering together. We used to do it a lot more frequently. Now it's a little less infrequent. We're on like the high school reunion timeline plan, but that's a good thing, right? Because it means that we're doing a good job. We're still making updates on those long-term research, talking with our industry partners all the time, our government partners. And again, it's all about this continuous improvement, right? As soon as you stop improving, that's when you get yourself in trouble. And that's the whole point of root cause analysis. It's really that long-term prevention and then staying really alert based on it, right? So I think Felice mentioned, whether it be 2 a.m, they're ready to go out and do what they need to do. So in summary on this one, ultimately, we didn't really find a smoking gun. It was a lot of smoking guns. There were several different things. And really, I think what we came out with our root cause analysis in this 20-year process was really recognition and understanding that Salmonella Newport is in the environment. It's in our waterways. It's in our raw poultry, these different things that we identified. Fine, it's there. We get that. We don't grow food in a bubble. But what we needed to do was really embrace this food safety culture of prevention, right? Continuously looking at it, learning things from research, learning things from, of course, our struggles where we trip and have an issue, a recall, an outbreak, to implement these multiple hurdles to minimize introduction and growth. That's what we're trying to do. We're trying to prevent introduction. And then, of course, we definitely do not want to give it an opportunity to grow. And so we feel like with a lot of the best practices that we've implemented from these different root cause analysis endeavors, that we've gotten to a pretty good point. I'll never say a great point because I just can't. It's just bad luck. But I say, knock on wood, this process is never over.
Good afternoon. I'd like to take some time. I plan to take some time to build on the process that Vivian was talking about on root cause analysis, and then pull from some of the great examples that Felice and Laura provided, because this is really unintentionally, it's aligned just because that's experience with root cause. Mark Mormon talked last week about how FDA is differentiating root cause investigation with root cause analysis in our own internal procedures. The reality is that, as Felice presented in her example, these things are going on at the same time. So the investigation is how we're collecting information, how we're looking at potential sources, and we're doing our data collection. And then the root cause analysis is where we're using tools to help us to evaluate that data, structure our data, and come up with solutions. In practice, my experience is when we do the root cause analysis of the data we've collected, we might see other areas where we need to do a deeper dive and go back and collect some more data. So these two are really interconnected. But what I want to talk about first is what some of the limitations or some of the challenges are, some ideas about maybe how we can tackle the challenges, and then talk more specifically about some tools and how those might be applied, and then end off with some learnings. One of the challenges we have as a regulatory agency is when we're investigating an outbreak or an issue, these are often conducted after the outbreak or issue is over. I remember being involved on the industry side in an outbreak where we were informed by CDC and FDA. When we took our actions at the time and we started our investigation, but later on when we looked at the outbreak curve and we looked at where those cases were, it looks like we initiated our investigation long after the outbreak was over. And this is true for a number of reasons, because there's incubation periods for pathogens. You know, listeria can be 70 or more days before illness occurs. There's the time to link together maybe these cases and determine that there's a common source, and then evaluate what that source is or what that vehicle may be, and then initiate the investigation. So a lot of time we're looking in the rearview mirror. Likewise, when we're looking as a firm, if I'm looking at an issue, if I'm responding to a failure of a system or spoiled product or listeria in a sample or a consumer complaint or a customer issue, or heaven forbid a call from CDC and FDA, these are also often retrospective for the same reasons. I'm acting on verification information that's after the fact. Analytical testing, another factor is analytical testing. When we're conducting testing, particularly of ingredients and products, that's statistically limited. And then there's challenges even to the method's recovery rate if we're relying on that method to pull something out of another type of sample, like an environmental sample. And I'll talk a little bit more about that. Challenge with the produce industry, in particular on the fresh side, is that we have short shelf lives. As I mentioned, often it's after the fact that we're figuring out there was an issue. I might not have product left anywhere to go back and sample. I'm certainly not going to have large volumes of this sitting in a freezer somewhere or in a warehouse somewhere that I can go back to if it's a few weeks later. So that short shelf life often limits our ability to, in real time, evaluate the scope of an issue or characterize an issue in a particular lot of product or ingredient. Another factor or another challenge is that where we have multi-component products like a salad mix or an assembled meal, supply chain is often complex, and the sophistication of our suppliers may impact our ability to do an investigation, how good their data is, and how aligned their systems are to what we would expect, or even their cooperation where we're doing that investigation. Sometimes a manufacturer might even source one material from a variety of suppliers, growers and suppliers, and this may make attribution of a particular supplier or material to the implicated lot very difficult. And this is where the importance of traceability, scope, accuracy, and precision are very important. Many processes are very complex. I'm going to talk about this next, but some other issues could be that for some agents, we have limited tools. We might not have good sampling methodology. Maybe it's not validated for a certain matrix. Maybe we don't have good indicators for that pathogen. So we can look at other data to help us to determine risk. And sometimes we just might not have in our records or available sufficient information to evaluate underlying causes. Next slide. So this is a produce example of what I was sharing with you earlier. This is a cyclospora outbreak and with bagged salads. And here we see that the sampling, if you look at the blue arrows, that's where we started to sample. So that's where we knew there was an issue. And so we started to sample fortunately at the tail of the outbreak, but the outbreak had already happened. So whatever the failure that occurred had happened in the past. And what we need to think about in these examples is if we're going into a firm, if we're going into the field, if we're looking at a supplier or a distribution center, we need to not just be looking at what's happening at the time we're there, because maybe everything's working then. We need to think about what could have happened in the past in this operation that could have led to a failure at some point in time that contributed to the risk. So we're always often in the position where we're having to look retrospectively and having to think beyond the checklist or the inspection or the audit and think about the systems and their robustness and what failures could have occurred. Next slide. Many of you may have seen this when we were talking about COVID controls. I'd seen this before that from the airline industry and talking about some of the failures that might occur in aviation and when disasters occur. The reality is that in a lot of cases, and produce is a really good example, we have a variety of factors that we rely upon to control a particular hazard. A lot of systems need to work. We don't have a pasteurization of the ice cream mix, and we know that everything before that's not an issue and just have to focus after the pasteurization. We're looking at a lot of factors in the environment, in transportation, in food worker handling, in suppliers, in water, in harvesting equipment. All of these things could have factors that could contribute to risk. And in the version I think that you'll receive after the fact, I've got some more details on each one of those categories. But either intentionally or unintentionally, we're looking at something like Swiss cheese. I might have either by design a multi-hurdle process where there's several factors that control risk. Maybe those things not complete, each control don't completely manage the risk, but together they manage risk to some degree. Or maybe just incidentally or not deliberately, we have some barriers to a hazard coming through just in the nature of a product or process. It's only when things line up, the holes line up in each of those barriers that we end up with an issue. And if we think about that in the context of figuring out what happened retrospectively, that creates a challenge because some of those failures may have been around for a long time, but the hazard wasn't coming through because something else blocked it. Next slide. Another challenge we have is analytical testing. Many of you have seen this before. This is one of my go-to slides in presenting to management and folks that are relying on testing in isolation. Here you can see in the table, so testing really only tells you what's in the sample we're evaluating. And it can really only, if we're sampling a lot, can really only detect something significant and not really very good at low-level sporadic contamination. And if you can see in the table at the bottom grouping, there's N equals 60, which is a pretty robust or one of the more robust sampling plans that we would use. And certainly if 1 in 10 units is contaminated, I have a pretty good chance of detecting that lot's contaminated. So that's a 99.8%. I still have 2 in 100 chance of missing it, but most of the time I'm gonna catch it. But even if you go down to 1 in 100, I have, if you look to the column, you can see 57% of the time, I'm going to accept that lot as being okay, even where 1 in 100 units are defective. So you can see just relying on testing in isolation isn't really going to tell you whether or not there's an issue or a sporadic issue. Certainly, if there's a positive result, that's great. If there's a negative result, that doesn't necessarily tell you there wasn't a problem. And the lesson for me is always that we have to be looking at other factors other than just test results and really understand other information about a food system or a process in order to evaluate this data in context. Next slide. We also have the challenge of our ability of variability in the environment and variability, the difficulty in isolating maybe the target of concern in an environmental sample. And here I use water. We see the presence of a hygiene indicator does not necessarily correlate with the presence of pathogens. So in one outbreak in Yuma, it was related to the water source, but the generic E. coli was within what would be considered the specification for that water. So it didn't necessarily indicate in isolation that there wasn't a problem. Also, we know that pathogens, if they're present in the sediment, might not be recovered by water testing. And we also know that the microbiological profile of water will change based upon a variety of factors. Again, testing is great, but all needs to be taken within context. There are some ways, next slide. There are some ways to concentrate water and get a better sampling. This is the dead-end ultrafiltration system that FDA uses for water analysis. And so it is able to target and isolate a pathogen out of a large volume of water. So it can get us closer, but still we need to take this data in context. Next slide. We know also that genomics, so whole genome sequencing, genetic analysis can give us information about pathogens that can help us to determine relatedness or commonality, or whether something's a recurrent issue in a facility or an operation, or whether it's something that's unique. This is just an example from an outbreak where we tied together cases of hepatitis A, and shows how whole genome sequencing allowed us to identify a cluster and then start to search for a root cause. Next slide. Traceability is also important. I mentioned earlier, in particular, where a company would have a complex supply chain, or where we're trying to understand what the root issue is of an issue or an outbreak. And this is an example, again, for hepatitis A. And you can see we had pretty good precision. We were able to narrow it down to 2 distributors, but we, in the end, only got it down to 6 growers. And that's pretty precise, but that doesn't tell us exactly what grower we need to focus on. So the greater precision and effectiveness of traceability, the more effective our root cause investigation can be, because we know when where to target our efforts. We were recently looking at an outbreak investigation, not related to produce, smaller operator. They had no records. They have a variety of suppliers for a particular material, and it was very difficult for them or for us to even know where to look on the supplier side, because there wasn't information about which supplier provided materials for the particular lots in question. So traceability is critical. Next slide. And just a reminder that we have a traceability rule, and the idea of this rule is to target higher risk materials, some of them are produce related, and establish supply chain requirements to help us with this problem. Next slide. We talked about some of the challenges, and certainly there are some barriers to overcome. These can be dealt with just through taking information in context, and also, as Felice had mentioned, and in the example Laura gave, having an interdisciplinary team to provide information on where to look and what to look for. Luckily, we know for the pathogens, if we know the target, we know what those areas are that could be of concern just through experience, but those can help us to focus and interpret the data. I want to just take the last couple of minutes and walk through again some of the tools that are in place or that exist for root cause analysis. Next slide. So the first step would be to brainstorm information that areas that we need to look at, and what our theories are of what could have happened. This needs to be a multidisciplinary team. This needs to include people that have a really good understanding of the process and understanding of the operation, so that they can provide the right insight. Next slide. We've heard in several examples the use of the fishbone diagram. This is not magic. This is just a way to sort data and make sure that we're evaluating all of those areas that could be of interest for a particular hazard or particular process. This is an example, and I stole this one from Natalie Dyenson for a dole outbreak that she was providing information on. And I can show you that the next one is for the next slide. So this is a much more complex one, and you can sort through the animation. You can click through the animation on that. This is a more complex one that applies to a more complex process. So you see, this can help us sort our information. It can help us make sure we're not missing something and help us rule things out. Next slide. You can just click through all of the animation there. Thank you. Next slide. There is a 5Y process, and you can click through all of these. Mark Moorman presented one on Listeria when we last spoke. This is another one related to Salmonella. And the issue that we were digging into is, why was there Salmonella? It's because was present in overhead irrigation, because the irrigation treatment wasn't sufficient, because the delivery wasn't sufficient, because it wasn't validated. And digging that deeply can get us to, next click, what the corrective action would be, which is to work to validate and verify the delivery of the chemical in the system. This is one where we need to go through and validate as we do our deep dive to make sure we're on the right path, because you can go a number of directions on 5Y analysis. Next slide. This is an example of where these were merged. So this was developed for use in the Food Safety Summit by Tim King, and it just hybridizes the 5Y with the fishbone, the questions there. So there's no magic bullet. There's no required way of approaching this. It's whatever works for what you need to analyze the data. Next slide. And as I mentioned, there's a variety of tools. These are all different statistical tools. Any tool that can help you evaluate data to get to the information that you need is appropriate. Next slide. If you can click through all the steps here, I can just talk about this as the last process I want to cover. There is something called a Go See Think Do. It's part of the Toyota manufacturing process, or DMAIC. This is just a process to help you sort through where an issue occurs. It's not unique to food safety. It says, did we identify an issue? Is the root cause already known? If yes, of course, we take actions. If not, then we understand how the problem happens. We ask if there's a standard. Yes, is the standard being followed? No, that makes it easy. We follow it. If yes, then we identify root cause. We implement solutions. And next click, we update the standard. And then we verify that the new approach is working. Last slide, please. As the previous speakers mentioned, the outcome of the root cause analysis is going to help to hopefully identify a failure that led to the issue. Hopefully we identify underlying factors, if we go deeply enough, that led to the failure. And it hopefully also provides us information to characterize and determine the scope of the underlying factors and inform our corrective and preventive actions. But often there's no smoking gun found. And in the two examples that we heard today, there wasn't a smoking gun found, but those samples identified areas that needed to be reinforced and potential factors that needed to be reinforced. And in the case that Laura presented, research and ongoing verification was needed to further understand what happened. Sometimes it's later on, after we've started up and we've been collecting data or done additional studies, that we really identify those areas that we need to address. Just because we don't find a root cause didn't mean we failed. With that, thank you.
Thank you to our speakers again. Really appreciate it.
Thank you.